Hi, my name is Jason Gill, and I'm a member of the Center for Phage Technology at Texas A&M University. In this video, we're going to talk about the Center for Phage Technology's Galaxy and Apollo platform for whole phage genome annotation and analysis. First, we'll start off by talking about what is Galaxy. Galaxy is not an analysis tool in itself. Galaxy provides a platform for performing reproducible bioinformatics research. It provides a web browser-based user interface for other command line tools. It provides the history of actions performed and saves your tool outputs. It allows users to chain operations together into workflows to perform very complex analyses. Finally, Galaxy is an open source, meaning free to you, platform with an active user community. Galaxy operates by interfacing with any Linux command line program by a short script, which is called a wrapper. The wrapper represents the input options to the user and passes these properly structured commands back to the program, such as, for example, BLASTP or Glimmer3, or any other command line tool that you may be interested in using. Galaxy runs that job and keeps a record of that job. It then keeps track of the inputs that were sent to it, and it keeps track of the outputs that are produced, and which are all placed in a user history. Ultimately, Galaxy offers the power and flexibility of command line bioinformatics to the average biologist just by using a simple web interface. So why use Galaxy? This is a simple diagram illustrating some of the benefits to the user for using Galaxy. On the left is the user, which is you. And what you see is just the web browser interface for Galaxy, and what you don't have to see or manage are the server clusters, the cloud computing infrastructure, or the complicated command line interface and file management for your tool inputs and outputs, as those are all managed and organized by Galaxy. Why use Galaxy for phage? Galaxy is a popular and well-supported bioinformatics infrastructure. There are over 125 Galaxy platforms currently running around the world which are publicly accessible. Galaxy automates tasks and retains inputs and outputs for future reference. It's customizable for individual use cases, and the user retains control of the analyses. It has uses in education where students can see and interpret their tool outputs and customize the analyses themselves, which is an advantage over the fully automated annotation platforms that are available. It's useful for novel or unusual genomes, which will permit the user to customize or modify their analysis approaches. It also allows the user to go beyond annotation to conduct comparative genomics, mutation analysis, and phylogenetics. This is a simple overview of the general Galaxy interface. On the left panel, there is a list of tools which are available to the user. The center panel is dominated by the analysis and results. This is where, when invoking new tools, options will appear. And this is where results can be displayed when the tools have finished running. The right panel is the user history, which keeps track of all jobs that are executed by the user in the order that they were run. This is an example of the general order of operations while working in Galaxy. The user uploads data on the right into the history of their Galaxy account. From the history, that data can be passed on to tools in the tool interface in the main window. What the user does not see is the tool interfacing with the wrapper, which invokes the command line tool or script. The output of the tool is then returned to the history. This process can be repeated as many times as needed for different analyses. At any time, the user can then download the results of any individual analyses or data sets to their local computer for analysis on their own machine or elsewhere on the internet. This is an overview of what the history looks like in Galaxy. The history panel contains all input and output for your analysis. All of the input data for tools, such as sequences or GenBank files, have to be uploaded into the history in order for them to be used in Galaxy. Tools in Galaxy can only access data when it is in a data set in the history. All running jobs appear in the history in the order that they were entered. All output data will also appear in the history. Each item in the history is called a data set and appears in the order that it was entered. Each item is numbered. The numbers can't be changed, but the names can be edited by the user if desired. Gray items seen at the top of this window are queued to run, which means that they've been entered by the user but have not yet reached the command line interface yet. They may be waiting for processing time from the server, or they may be waiting for a previous job to finish if they require that tool's output as their input. Yellow items have been passed off to the tool and are currently running. The amount of time it takes for them to run is dependent on the tool which is being executed. Simple scripts might execute in only a few seconds. Certain analyses which are more intensive, like BLAST or interpro scan, might take several hours to complete. Green items are jobs that are completed and ready for viewing. They can be downloaded by the user, or they can be input into the next tool. Red items are jobs that failed to run or returned an error. In the case of red jobs, the user can investigate the bug report which is generated to determine if it's a user error or a problem with the system. The user can create an unlimited number of new histories to keep track of related analyses. The window here shows the history browser window, where the user can sort through and manage all the histories they've generated. Datasets can also be copied or moved between histories, and they can also be copied or shared between users in the same Galaxy instance. Analyzing data in Galaxy. The image on the left shows the input wrapper, and this is the wrapper for the common tool BLAST-P. This wrapper has all the same options that are available to the user in the command line, but in this case they're presented on the web page in a graphical interface. The tool options are determined by the wrapper, but these are all switches that could be entered at the command line. The input data for this tool, like with all other tools, must come from the history, so it must have already been uploaded to the user's Galaxy account and appear there as a dataset before it can be used for other analyses. The output of this analysis will appear in the history when the job is launched, shifting from gray to yellow to green. Workflows. One of Galaxy's most powerful features is the ability to connect jobs into workflows. Some analyses take a long time. Rather than waiting by the computer for one job to finish to execute the next one, the output from one job will automatically be handed off to the next when it finishes. Shown below is a simple workflow. This workflow will take an input genome file, which is in the GFF3 format, that contains the annotations and DNA sequence. The workflow will export the entire genome DNA sequence and run it in BLAST-N. At the same time, it will export all the DNA sequence regions and protein coding genes, translate all of these sequences into protein sequences, and then hand those off to BLAST-P for analysis. Each tool is configured before the workflow starts, so it can be left to run once initiated. The results of each step are saved in the user history and can be retrieved later. Workflows are facilitated by the wide variety of parsers and converters that are available in the CPT Galaxy. Some of these are shown here in this window, for example, tools for exporting protein coding genes and for translating DNA into protein. WebApollo. WebApollo is an interactive genome visualizer that supports collaborative genome annotation. It is an extension of the popular JBrowse genome viewer that allows editing of the genome inside the web interface. It is described as a similar concept to Google Docs, but for genomes. WebApollo maintains user annotations and multiple evidence tracks to guide annotations. The CPT has developed tools that bridge Galaxy into Apollo, which are now seeing uses on our Galaxy installation and on other installations around the world. This illustrates the linkage between Galaxy and Apollo, which is often not that clear to new users. On the left, you have Galaxy, where the user can upload their data and conduct their analyses, and they can, if desired, be downloaded to the user's computer. On the right side, you have Apollo, which is essentially an extension for JBrowse and allows for evidence review and annotation. While each user's account in Galaxy is linked to a parallel account in Apollo, each system maintains its own separate database, Galaxy containing user datasets, and Apollo containing user organisms and annotations as JBrowse instances. What we need is a way to link the analyses conducted in Galaxy to organisms in Apollo, and for that we generated the Galaxy-Apollo Bridge. It includes tools that can generate and modify organisms in Apollo, and transfer these tool outputs from Galaxy to the Apollo database, where they will then appear as new evidence tracks. It also includes features to retrieve data from Apollo back into Galaxy, where they can be subjected to further analysis. The suite of tools are referred to as the Galaxy-Apollo Bridge. These tools are generally used as part of workflows that conduct complex analyses and transfer data to and from Apollo, and are not typically executed by the user as a standalone tool. The user annotations in Apollo, appear at the top of the user-created annotations track of display, shown in yellow. This top bar is the only track that is actually editable by the user in Apollo. Evidence tracks, which are in the white field below, are generated by various tools in Galaxy, and they are imported into Apollo via the Galaxy-Apollo Bridge. Each organism has only a single user-created annotations track, but it can have an unlimited number of evidence tracks below it, which can display the outputs of any kind of analysis that can be mapped onto a DNA sequence. This schematic shows an overview of what the annotation workflow looks like on the CPT Galaxy-Apollo system. An unannotated DNA sequence is uploaded into Galaxy and then subjected to a structural workflow to identify genes. The organism sequence and tool outputs are then transferred to Apollo, where users are able to call genes in the user annotation track based on the evidence. These user annotations are then transferred back into Galaxy, where the called genes are analyzed by additional tools, and these are then transferred back into Apollo, where the user can then use this data to annotate their gene functions, again in the user-created annotations track. When they're finished, the data can be retrieved back into Galaxy for export as either GenBank or GFF3 format files, for additional analysis, or for submission to sequence databases. So part of the process is conducted in Galaxy, which is where the actual data analysis occurs. Then the actual user annotation occurs in Apollo. All of this work is facilitated by transfer steps, which are accomplished by the Galaxy-Apollo Bridge. If desired, the user can continue to run other analyses in Galaxy and import these tracks back into Apollo for as many cycles as they desire. For more information on using the Galaxy platform, we encourage you to review the extensive Galaxy training resources that are available online. These include those available at the main Galaxy homepage, Galaxy 101, and of course the Galaxy training materials produced by the Center for Phage Technology, which are specific to our system and located at cpt.tamu.edu/training materials. The CPT training materials contain detailed instructions on how to upload and manage your genome in Galaxy, and how to run the major analysis workflows. If you use our system, we also encourage you to cite our publication in PLOS Computational Biology that describes the platform and the associated workflows. The CPT Galaxy-Apollo platform is supported by the National Science Foundation, Texas A&M AgriLife Research, and Texas A&M University. We would like to thank the many programmers and scientists who have helped design and build the system, and the excellent and supportive Galaxy and Apollo user communities.